Alright everybody, this is going to be the video for chapter 44, which is on osmoregulation. I'm mostly just going to go over the kidney stuff for this one because that's really the bulk of it. I'm not going to so much go over the organismal physiology, so like the stuff about like sharks and insects and the different like types. I'm not going to get into that. I'm just going to go over mostly the nephron. So to start, I just want to mention, so this is the nephron. Nephrons are the functional units of your kidneys. And your kidneys are composed of two sort of regions. You have your renal cortex and your renal medulla. The renal cortex is sort of around the edge of the kidneys, and then the renal medulla is sort of the inside. So you can think of it as the cortex is sort of the covering of it, and then underneath of that, once you peel that back, you see the renal medulla. And all I really want to mention about that is the renal cortex is going to be everything above this dashed line, and then everything below this dashed line is going to be the renal medulla. Alright, so to start off, just what does a nephron do? What's the point of the kidneys and osmoregulation in general? Well, the big thing is it filters blood. That is number one. Its main function is to take our blood and determine what our blood needs more of, needs less of, and what we can get rid of. And the stuff that we get rid of is going to become urine, which will eventually go to the ureter and then the bladder, and then you'll pee it out through the urethra. So this all starts by blood coming in. So right here, we have an artery that is coming into this first component of the nephron. This is called your afferent artery. And what it's going to do is it's going to come in here and it's going to go around in this really complex sort of labyrinth of capillaries. So all of these capillaries are going to allow for exchange of various things. And then once a lot of stuff goes out into the nephron or filtered, the remaining bits will come out of this artery, which is our efferent artery. So this is the efferent artery and this is the afferent artery. And so this whole first step is called filtration. So what just happened was filtration. And filtration means that the blood goes into this structure and a lot of it gets filtered out to continue through the nephron. So let's start by naming this first structure. This first structure here is the glomerulus. So the glomerulus is going to be the site of filtration. So to start, let's just talk about what gets filtered out. So what is going to be continuing through the nephron? Well, we're going to have a lot of H2O that's going to keep going through. We're going to have ions like sodium, potassium, and chlorine. And we're going to have amino acids or proteins. So that's what can go into the nephron. So what does not go to the nephron? What's coming right back out through this efferent artery? Well, we're still going to have a lot of our plasma, which is going to be a lot of water. So not all of the water is going to go through here. And then we're also going to have our red blood cells not get filtered. Those are the primary ones. All right, so we have filtered out a lot of substances. We filtered out all these solutes, we filtered out water and amino acids. So what's this next sort of section that it's going into? So this is going to be our proximal convoluted tubule, which gets shortened to PCT for a while. So that's this next segment. We're still in the renal cortex. And so the big thing that I want you to remember about the proximal convoluted tubule is that this is going to be about 70% of nutrient reabsorption. And so let's talk about that word reabsorption. So reabsorption means that we are going to take stuff that we are filtering out and we're going to pull it back into the interstitial space, which can then make its way back into the blood. I didn't draw it up here because it would look really cluttered and confusing, and it already is cluttered and confusing. But all around your nephron, you still have arteries and veins. So you have blood vessels running very closely to all of this. And so what we can do is we can move things that are in this lumen, the lumen of the nephron, we can move it out here so that it can be picked up by a blood vessel. And that's called reabsorption. So remember, filtration was going from the blood into the nephron. Reabsorption is going from the nephron back into the blood. And so our proximal convoluted tubule is going to be about 70% of that nutrient reabsorption. So it's the very bulk reabsorption. And this is especially easy for it because it has all of these tiny hair-like structures, which are called microvilli, which you may remember from the digestion unit. And these microvilli are going to be really good at pulling nutrients back. So I want to just kind of blow up one section of this proximal convoluted tubule so we can see the various channels that we are dealing with. So the first thing I want is just to get ourselves oriented. So first we have the lumen. So the lumen is where your pre-urine is. So this is where all of this is going. And that's the inside. But then we don't just have one membrane, we actually have a second space, which is going to be epithelium. So we have this middle space between the lumen and the interstitial fluid. So the majority of things are going to be in this interstitial, or in the urine, in the lumen. And then we're either going to be able to move them to the epithelium, and then once things are in the epithelium, then they can get out and go back to the interstitial fluid, which can be reabsorbed. So it's important to understand that there are three compartments. We have the lumen, the epithelial space, and the interstitial fluid. So the first channel that we need to talk about is sort of the most important one, and that's this one right here. This is your sodium, potassium, ATPase. So I don't know if you guys can see things there, but this is the sodium, potassium, ATPase, or NAK pump. And hopefully you remember from last term your active transport, primary and secondary. So this is going to be primary active transport. Let's write that down. Primary active transport. And so what this is going to do is it is going to take sodium from inside the lumen or the epithelium. So it's going to take 3 sodiums and it's going to move them out into the interstitial space. So these 3 sodiums can now be reabsorbed into the blood. So now we have Na out here, but then it's also going to bring 2 potassiums into the epithelial space. So we have 2 potassiums coming into the interstitial space. The problem is both of these molecules, or both of these ions, are actually moving against their concentration gradient. So right now, as it stands, we have a high concentration of sodium outside the cell, and we have a high concentration of potassium inside the cell. So we are not moving from high to low, and anytime you're not moving from high concentration to low concentration, you know it's going to be active transport. But in this case, so we need to get energy somehow. We need to harness some energy from something, and what is that? That's going to be ATP. So we're going to convert ATP to ADP, and that's going to generate the energy to move this sodium out into the interstitial space to maybe be reabsorbed, and it's going to move this potassium into the epithelial space, which would technically be secreting. So this is the last word that we need to talk about, is secretion. Secretion means we are moving things from the blood to the lumen. So it kind of means the same thing as filtration. It's just we're secreting things into the urine, which will eventually then be waste as we ATP it out. Okay, so that is the first big important pump to understand is your sodium-potassium ATPase, because this gradient that we've created where we now have really high sodium outside the cell is going to be extremely important for the rest of the channels. So the first channel, and the one I want to talk about here in the proximal convoluted tubule, is going to be your SGL2, and that's this. So this is really the only example you need to know, SGLT. And this is going to be a sodium glucose symporter. So this is going to be a sodium glucose symporter. And if you remember what symporter means, that means that this is secondary active transport, and we're going to be moving two things in the same direction. So remember in our lumen, we had a bunch of sodium. We also kept, I didn't say it up here, I apologize, but we also keep glucose in our filtrate. So in this initial filtration step, there's also glucose running through this. But let's say that we want that glucose to end up back in our blood, which is going to be over here in the interstitial space, right? So we also have glucose. This is going to be beneficial to have these sodium-potassium pumps because it created a really low sodium gradient here in the epithelium. So we're going to erase this now. Hopefully you understand your sodium ATP, or your sodium-potassium ATPase. So the effects of that sodium-potassium ATPase is we now have a low concentration of sodium in this space. So that means that now the sodium in the lumen, in the urine, can naturally flow down its concentration gradient into this lower concentration of sodium. And it can do that because we had the sodium-potassium pump creating the gradient. So if you ever see a question that says what creates or what maintains the sodium gradient, it's your NAK ATPase, or your sodium-potassium pump. So anyway, now that we have this sodium going through this SGLT, sodium glucose symporter, glucose can sort of hitch a ride. So glucose is high in this lumen, and it's low out here in the epithelium. Or sorry, I said that backwards, I apologize. Glucose has a relatively low, because ultimately, you know what, we're not even going to get into this. Just know that glucose can essentially hitch a ride with this sodium and enter the epithelium. And it can do that because this sodium is moving down its concentration gradient, which no longer requires energy, but it did require indirect energy because it needed this sodium-potassium pump. And then the last two channels I want to talk about here are a lot easier. You have a GLUT transporter, and we have a potassium channel. So let's say that we pumped this potassium in to the epithelial space, but we might want to reabsorb that potassium. We might want to keep that potassium. The K+ channel allows potassium, which has now have a high concentration in the epithelium because of this pump. This potassium can now just kind of leave on its own, and the glucose that we've pumped into the epithelium space can now move out through these GLUT transporters. And so that's it for the proximal convoluted tubule. The big takeaways here, I just want you to know about this sodium-potassium pump and understand that that maintains the sodium gradient. Excuse me. So remember, proximal convoluted tubule, so we've done the filtration, we've done the bulk reabsorption. We're now going to dip down into the renal medulla region. And this whole structure here, this big like loop, is the loop of Henle. And it's sort of divided into two parts. It has a descending limb, and it has an ascending limb. So this is the descending limb, the part that's going down, and then the ascending limb is this portion that's going up, and it has this sort of thicker portion here. But the whole thing is called the loop of Henle. I'm going to erase this. So the loop of Henle has these two portions, and they do two very distinct things. So the first thing in the descending limb is that it only moves water. It doesn't do anything else. So the only channels present in the descending limb of the loop of Henle are aquaporins. So all of these little spaces are aquaporins where water can be reabsorbed. So water can now go out, because remember, in this proximal convoluted tubule, we sort of created a lot of ions out here. You know, we were reabsorbing things like sodium and glucose. We reabsorbed some amino acids. So all of these solutes out here that we created from this proximal convoluted tubule are now going to be able to pull water back. And that's through osmosis, right? Remember that water always moves to dilute substances. So if I moved all of these salts out here, you know, maybe we put some potassium out there, some chloride out there, all of that, now all of this is drawing water out of the descending limb of the loop of Henle. And that's all just about osmosis. It's all passive, and that is all the descending limb does. So the ascending limb of the loop of Henle is going to be NaCl transport. That's going to be the primary thing that it does, and it's going to do that through a specialized secondary active transporter. So I've sort of zoomed in again on this little portion of the ascending loop of Henle. So again, just to orient yourselves, we have our lumen here, so this is the urine on the inside. We have this middle space, the epithelial space, and then we have our interstitial space. And this is, remember, where reabsorption can occur. So this first thing right here is gonna again be your NAK pump, or your sodium potassium ATPase. And so that's gonna do its thing, which is going to create a low amount of sodium in the epithelial space. So we now have a low concentration of sodium here. And so when we have the sodium in the lumen, it's really going to want to flow out into this epithelial space through this channel called an NKCC2. And that stands for sodium potassium chloride cotransporter. You don't have to memorize the name of that, but I do want you to know the function. So this sodium moving from a high concentration in the lumen to a low concentration in the epithelial space is going to actually generate some energy, right? Because it's moving down its concentration gradient. And again, things can sort of hitch a ride with it. And in that case, in this case, it's going to be K plus and 2 Cl minus. So potassium can then enter the epithelium, and chloride can then enter the epithelium. And then once all of these substances are in the epithelium, they'll have specialized channels to allow them to enter the interstitial space and be reabsorbed. So loop of Henle, the descending portion, is all about water reabsorption through aquaporins. And then the ascending limb is mostly going to be NaCl reabsorption, so salt reabsorption, which remember we saw a lot of that up here in the proximal convoluted tubule. It's going to happen to a much lesser degree in this ascending limb. And so that's it for the loop of Henle. So the loop of Henle is going to come back up into here now, and this second to last portion is called your distal convoluted tubule, or your DCT. And for the DCT, I want you to mostly just think fine-tuning. So this is going to sort of be the last point before we enter the collecting duct, where we're actually able to sort of mess with the solutes. And so the solutes that we want to mess with, sort of the biggest two are going to be sodium and calcium. So we haven't talked about calcium yet, but this is going to be a point that allows us to move calcium to either be reabsorbed or secreted, depending on what the blood needs. And there is also going to be some chloride movement as well. So when you think distal convoluted tubule, just understand that that's sort of the fine-tuning point. So again, I've blown it up here. We have our interstitial space out here. So this is where reabsorption can occur. We have our epithelial space, and then we have our lumen. So our urine is up here, we have our epithelial space, and then we have our interstitial fluid. So yet again, we have our Na plus K pump, or the sodium potassium ATPase. So that's going to do what it has been doing and create high sodium in the interstitial space and low sodium in this epithelial space. So the two channels out here that are important are the NCC, which is called the sodium chloride cotransporter, which is going to move sodium and chloride into the interstitial space and eventually out into the epithelial space and eventually out into the interstitial space to be reabsorbed. But sort of the unique part of the distal convoluted tubule is going to be the calcium channels and the calcium exchangers. So we have calcium channels which can allow calcium to sort of passively flow into here. And then it's going to have, there's going to be another channel that is going to be the sodium-calcium exchanger. And so sodium-calcium exchanger is going to be, again, secondary active transport, but it's going to be antiport this time. So remember, symport means things moving in the same direction. Antiport means they're going to move in opposite directions. So remember, we have super high sodium outside in the interstitial space. So what this transporter is going to allow us to do is for this calcium to leave the cell by harnessing this sodium coming back into the epithelial cell. And so that's your sodium-calcium exchanger. So again, big picture, distal convoluted tubule is going to be a fine-tuning point. It's going to sort of try to get the urine and the blood just right and sort of find the best balance. And then this last portion here, where we dip back down into the renal medulla, is going to be the collecting duct. Let me make sure that this is in frame. It is. So the collecting duct is going to eventually lead to the ureters. And the ureters will eventually transport the urine to the bladder. And these are all outside of the kidney. I'm very much like showing this down and summarizing. And then from the bladder to the urethra, which is where the urine will be excreted. So excretion is the final step, and that is peeing. That's you removing the urine from your body. But anyway, back to the collecting duct. Once again, we have blown up the sort of this section here, and we have our lumen, which is where the urine is, epithelial space, interstitial space. $5 if you can guess what this one is. It's the same thing it has been. It is an NAK pump. And it's going to do what it has been doing this entire time. And then the only sort of special things about the collecting duct is it has these channels that are referred to as ENaC, which is just a fancy word for epithelial sodium channel. So this is going to allow sodium to enter the epithelium, and then it is also going to have potassium channels, which allow potassium to enter here. Very importantly, these are both forms, oh, I apologize, I said this incorrectly. This potassium channel is actually going to move potassium into the urine to be secreted. So if we have extra potassium in our blood that we need to get rid of, we can pump it into the collecting duct and it can enter the urine. Both of these two channels and these processes, this sort of last stage of sodium reabsorption and potassium secretion, are controlled by the hormone aldosterone, which we're going to talk about after we get through the anatomy here. And then the last pump sort of transporters that we have in the collecting duct are aquaporins. So this is again an aquaporin, which is going to allow water to be reabsorbed. H2O. And so that is it for, well actually there's one more element of the nephron I want to talk about, but that's sort of the gross anatomy of the nephron. The big things to remember is in the glomerulus, we have our afferent artery coming in and filtering the blood through all of these capillaries. And then the filtrate, which includes water, ions, glucose, and amino acids, is going to go through the proximal convoluted tubule, and these microvilli are going to help nutrient absorption, so this is bulk nutrient absorption. And then the filtrate, or the preurine, is going to go down into the descending limb of the loop of Henle, and in this descending limb, water is going to be able to flow out through aquaporins. So then it's going to go into the ascending loop of Henle, which was where salt reabsorption is happening. Let's write that down. Salt reabsorption. And very importantly, in this ascending limb, there is no aquaporins. I should mention that as well. So now we had some extra salt reabsorption in the ascending limb of the loop of Henle. Then we're going to go to the distal convoluted tubule, where we're going to sort of fine-tune our ion concentrations between the urine and the blood. And then it's going to go down into the collecting duct. The other thing I want to mention about the collecting duct that I forgot about is it also manages blood and urine pH levels. And it does this with these two little transporters. So this is a type A intercalated cell. So type A intercalated cell, and then you just have a type B intercalated cell. So type A intercalated cells secrete acid, and type B intercalated cells secrete bases. So when we say secrete, remember secrete means that we're going from the blood into the tubule. So type A is going to move acids in, and type B is going to move bases in. So this is going to help control the pH of your blood, which is really important. So then in the collecting duct, we also have our final step of sodium reabsorption, potassium secretion, and water reabsorption, which we said are sort of controlled by the hormones aldosterone for sodium reabsorption and antidiuretic hormone, or ADH, for water reabsorption. And then we have our urine, and it goes through the ureters into the bladder to be peed out through the urethra. So the last thing I want to talk about is actually at the very beginning here, and it's this green label where I had the JG apparatus. So JG stands for juxtaglomerular, I think. apparatus. And so this is actually going to be something that is surrounding the arteries. And its main functions are going to be to sense salt levels and to sense blood pressure. And so the portions of the JG apparatus that do each of this is the macula densa, so the macula densa can sense NaCl levels in your blood. So it can see how salty your blood is. And then in your blood pressure are going to be sensed by JG cells, which have what are called baroreceptors. So baroreceptors means parts of these cells that can sense pressure. So it's going to be able to sense blood pressure. So that's sort of the structure of the JG apparatus. Just remember it's surrounding the arteries. So let's talk about what happens when this macula densa and these JG cells sense low levels of blood pressure or low salt levels. It is going to start what is called the RAS pathway. And the RAS pathway, it stands for renin, angiotensin, aldosterone system. Right? So RAS pathway is renin, angiotensin, aldosterone system. So the first thing that's going to happen in this pathway, so step one, is you're going to have low blood pressure or low blood NaCl. So you're either going to have very low blood pressure or you're not going to have enough electrolytes. And remember, this low blood pressure is sensed by the JG cells, and the low levels of NaCl is sensed by the macula densa. So step two, the first thing that's going to happen is these JG cells are going to secrete renin. So renin is the first hormone in the pathway, and what is renin going to do? Renin is going to go seek out these cells or these hormones called angiotensinogens, and these come from the liver. So in our bloodstream, the renin is going to find these angiotensinogens and it's going to convert them into the second hormone, angiotensin 1. So renin secreted by the JG cells is going to go find angiotensinogens and convert them to angiotensin 1, which is then going to get converted into angiotensin 2 by an enzyme called ACE. So far we had low blood pressure or low levels of NaCl, and then we had the JG cells secreting renin, which convert angiotensinogens into angiotensin 1, which converts into angiotensin 2 thanks to the enzyme ACE. And so angiotensin 2 has a lot of effects. I'm going to erase this and sort of reset here. So now we have, at the very end, we have angiotensinogen, or angiotensin 2. So one thing that angiotensin 2 is going to do is vasoconstriction. So if you remember from chapter 42, we talked about vasoconstriction a lot. Vasoconstriction is the tightening of blood vessels. Remember, one of the stimulants for this entire pathway was low blood pressure. And so one way we can raise our blood pressure back up is using vasoconstriction. So remember, vasoconstriction means we took a blood vessel and the smooth muscle around the blood vessel is going to contract, which is going to shrink the diameter of the blood vessel. And that means we have less space for the blood, and it's the same volume of blood in now a constricted space, which is going to raise our blood pressure. So vasoconstriction raises blood pressure. So that's one problem taken care of. But remember, the other issue was we might have not enough salt in our blood. So the next thing that angiotensin 2 can do is trigger the secretion of aldosterone. Aldosterone is the second sort of effector of angiotensin 2. You remember we mentioned aldosterone over here in the collecting ducts as ways to upregulate sodium reabsorption. So aldosterone can increase sodium reabsorption. So angiotensin 2 now has done vasoconstriction to help raise our blood pressure, and has also secreted aldosterone, which is going to allow more sodium to be reabsorbed into our blood. The last effect of angiotensin 2 is going to be ADH stimulation. So ADH, which is antidiuretic hormone, is the last hormone in this sequence, and it is going to increase water reabsorption. So it's going to pull more water back into the blood, which will also help raise the blood pressure, and it'll keep us hydrated. So this whole thing is kind of the response to dehydration or low blood pressure. And so the end thing was this angiotensin 2 that has these effects of vasoconstriction, aldosterone secretion to increase sodium reabsorption, and antidiuretic hormone stimulation to increase water reabsorption. And I think that is it. I think that's all I'm really going to talk about for nephrons and kidneys. I will see you guys in the next one.